Cool, so I'm going to start. My name is Fred Azevedo, as you may know, and my part of the tutorial will talk a little bit about cognitive neuroscience. Basically, I will have to, in 30 minutes, tell you everything that there is about cognitive neuroscience. And if you can, please contact me anytime during the course, or you can just send an email to this address. So I will start saying to you that there are two classic books of neuroscience that are introductory but definitely worth it. The first one is Principle of Neuroscience, the Kandel one, very small. And the Gazzaniga is more about cognitive neuroscience, and is also as big as this one. And it's interesting remark to say that Michael Gazzaniga was the one that coined the term cognitive neuroscience together with George Miller, a cognitive neuropsychologist, and when they were in the backseat of a taxi in New York. So basically and obviously, the term cognitive neuroscience is a combination of the terms cognition, which is the mental action or process of acquiring knowledge and understanding through thought, experience, and the senses, and the term neuroscience, which is the science of the structure and function of the nervous system. Basically, if you want to put this in simple words, is the science that tries to understand how the brain enables the mind. Just a little review from anatomy to function. I'm going to concentrate in the brain. So the brain is composed of cerebellum, brainstem, the encephalon, and cerebral cortex, as is depicted in the picture. But there is also the amygdala, the basal ganglia, the reticular formation, which are not depicted here. The cerebral cortex is the part that is a main player in cognitive neuroscience, basically in cognition. And it can be further divided in 4 to 6 lobes, depending on the author. So the 4 common lobes are the frontal lobe, occipital lobe, temporal lobe, and parietal lobe. But you can also find the limbic lobe and the insular lobe. The insular will be here after you make a hole there. And the cerebral cortex is composed of layers, basically 6 layers of neurons, and are also organized in a columnar fashion. And it's very useful to take the cerebral cortex in this, as it is, and opens it and displays in a flat map, like I'm presenting here. By doing so, you can see that there is interesting properties arrive, and one of these properties that you can see here is that functional areas, they tend to stick together. So if you do a scheme of the functional areas, you can see that the visual areas, they dominate the occipital part, and then you have the somatosensory areas, and then the motor areas, and it's color-coded. And so now, talking a little bit about the core cognitive processes, I'm going to be very briefly when I'm talking about perception, action, attention, and learning and memory. And also there are other topics that I'm not going to talk about but are as important as the other ones, like language, emotion. And so, starting with perception, there is an interesting process, an interesting property of the brain, which is that when the neuron input arrive in the lower order sensory areas, normally they dichotomize and they're sent to the association areas. For instance, in this case, you have the visual signal which goes to the parietal association cortex and also the temporal association cortex. And this property of dichotomization of of dual streams in the from lower order visual areas to upper higher visual areas seems to be a property of this of the sensory systems. It's not only in the visual system but also in the somatosensory and auditory system. And from the parietal and temporal association areas, they reach the frontal association cortex, and then the signals which are processed in this bottom-up manner, they can be, depending on the objectives, sent down and modulating the new inputs. Normally, the parietal stream is more associated with the processing of spatial features, while the temporal stream is more associated with the identity of the sensory object and also related features, for instance, the emotional valence. And the first stream was, the first hypothesis of the dual stream scenario was proposed by Mishkin and Angelider in 1982, I think. And they were studying the visual cortex in monkeys, and then they identified the what pathway, which is the parietal path, which is the where pathway, which is the parietal pathway associated with spatial processing, and the what pathway would be the temporal pathway associated with object identity and so forth. So this is Francisco already told a little bit about the visual processing. Basically, very simple. The information reaches the retina and then goes to LGN with the lateral nucleus, and then V1, V2, and here you can see the dichotomization that I was talking about. So there is the dorsal pathway and the ventral pathway. And it's interesting to note that in lower order sensory areas like V1, you have simple features, the processing of simple features like direction and orientation. And as you go up in the hierarchy, they associate in more complex features like complex shapes. And of course, the visual processing is not that simple, as I was telling. And here is a diagram made by Fellman and Van Es in 1991. And basically, it shows the visual areas at a time that were known to be functionally connected when processing visual stimuli. One consequence of this distributed hierarchical processing is that in the higher or high order areas, you are going to have areas specialized to process very complex objects, for instance, faces. And this is one study that I really like. It was made by Doris Sau and Freiwald. And here they show to two monkeys images of faces and objects, and then they identify the face patches of the monkey first. And then you can see here that this area is preferentially activated by the images of faces. And after that, they insert an electrode exactly in this area, and then they show again the stimuli, and they see that there is an increase in spiking activity, which is very selective to faces. So if you show faces, you have an increase, but if you say any other kind of category, the spiking activity is not that intense. And in the paper, they claim that the selectivity of neuron was 95%, which is incredibly high. So another thing is that of visual processing is that context matters. So, for instance, here in this figure, it's you can see that even though there are just 6 elements, 3 Pac-Man style figures and 3 angles, you interpret this immediately as 3 spheres and 2 triangles. In the second case, you have also just 3 spheres, but you think there are 3 spheres plus 1 direct line. And if you remove just the impression that there is another line here, you immediately decouple these images. And here also there is one tortuous line that is also an apparent effect, is an illusion. It's not real there, but our brain immediately interprets that. And we can also see the neural correlates of such illusions. Here in this experiment, the authors were recording from the receptive field of the neuron, which is this circle here, and then they make this rectangle of light move, and then every time that the border reaches the receptive field, there is an increase in the neural activity. Now the interesting part is that they put now another rectangle of light here, so there is basically no change in this line, no change where the receptive field is, but when they move the overlaid rectangle of light, when the imagined border reached the receptive field, you would also see a significant increase in spiking activity. And when you just showed half of these images, you wouldn't see any kind of activity. And another illusion that I like a lot is the color illusion. Here you cannot see very well, but I can show to you, and I'm going to show to you that this blue and this yellow are actually the same color. I just put some blank white boxes there to show to you that, oops, come on. Yeah, so it's hard to believe, but it's true. And similarly, and this happens because the blue and yellow background. The second one that I like is this, very probably you have seen that, is again, because of the apparent difference in brightness, your brain interprets this as gray or blackish and this as white, but actually they are the same pattern of gray, the same tone of gray. Yeah. So the second thing that I would like to talk about is action. Basically, action is also distributed in a hierarchical way. So the planning of the action starts in the premotor and supplementary motor cortex regions. And then together with the motor cortex, the cerebellum and basal ganglia, the plan of action can be transformed in a command to be executed. And then that is passed to the brainstem and spinal cord. And then the spinal cord innervates the muscles, and then the movement can be really executed. And nowadays, we understand a lot how the neural signal is processed in the motor cortex. For instance, some authors, they taught a monkey to move this lever to one of these 8 positions, and they showed that it was relatively feasible to decode to which direction the monkey was moving the lever, only based on the neural signals. And one directly application of this is brain-machines interfaces. So in this video, you're going to see that the author implanted a youth array, which is basically a matrix of electrodes like this one. And here is an image of a similar implantation. And they collect the neural data and then decode and pass this to a robotic arm. And basically, the function of the monkey is to move the robotic arm, get the marshmallow, and bring back. And you can see that he's not using anything else, exactly the signal of the motor cortex. And nowadays, this technology has been also applied to humans, so there are lots of patients which are already moving arms with their thoughts and using also the youth array. It's very similar. Yeah, he's thinking and moving there. This is the arm is just connected to a computer, and here they strategically and conveniently put this thing in front, but they have he has an electrode and a cable that you can see here that's connected to a computer.
When you turn his head, you can see the wires.
Yeah, exactly. Yeah. Yeah, you can see that the monkey sometimes moves the hand because this is part of the training. There is when the monkey is also trying to move the arm, sometimes he moves the hands, but there is another scientist, Miguel Nicolelis, basically he did something and then the way that he trained the monkey after the monkey was implanted was that in the beginning the monkey was training with a lever and moving avatar or robotic arm, but then he taught the monkey that he should keep the arms constrained and just use the thought, and then the same thing happened. So moving forward, another important cognitive process is attention, which is the process by which organisms select a subset of available information upon which to focus for enhanced processing. Basically, in simple words, what happens is that there is an enhancement of signal processing of things that you attend to, or space, or locations, and there is a inhibition of processing of the unattended stimuli, in very, very simple terms. So for instance, I'm going to concentrate in 2 kinds of attention, which is spatial attention and feature-based attention. Again, I am concentrating in the visual system because I don't have time to go for every sensory system. But there is also auditory attention and sensory motor attention and so forth. So in the case of spatial attention, for instance, you can fixate here in the middle of the screen, and you can select for any reason, a portion of the space to concentrate your attention to. And in this case, is this this region that is marked. And the other type of attention that I'm going to talk about is the feature-based attention. So imagine now that you want to select only the yellow objects in the picture, like this cab and these two signs. What happens is that your brain is going to boost the signal in those regions, and then it's going to be easier. Well, it's going to be easier to detect, let's say, in very simple terms again. In fact, things are a little bit more complicated than that. For instance, for spatial attention, yes, you have an increase in firing rate, but you have also, while in feature-based attention, you have not only an increase in firing rate, but also you have a sharpening of responses. And here in this example, what happens, you can see that there is small spheres that are moving, small circles that are moving in one direction 100% coherently, but when you add noise, basically you add some randomness in the direction of movement, and then you have, if this happens, you have a widening of the tuning curve, which is basically the response to a certain direction. And if you are paying attention to a certain feature, if you want to pay attention to this, to the direction of the movement, for instance, for the stimulus, with attention, what happens, there is a sharpening of the response. So you are basically removing the apparent noise of the stimuli. And here is a classical experiment that shows the neurocorrelates of spatial attention. And basically what the authors did was they taught the monkey to fixate centrally, and then they were recording from a receptive field that, for instance, is here. And then the stimuli that they showed was a classic Posner stimuli. Basically, the monkey is looking at this fixation point, and then there are two Gabor patches that are going to flicker, and at some point, one of the Gabor patches is going to rotate, and the monkey has to detect this change of orientation. And then you can prove that that there is an attentional effect when the monkey has increase in sensitivity. This is the behavioral curve of the monkey. Basically, is how easy the monkey can detect this orientation change when there is when the stimulus become easier to detect due to attentional effects. You can see that there is a shift of the curve to the left. And after that, if you put a record from a neuron, let's say in V4, like as they did, you can find that when the monkeys pay attention to the stimulus, there is an increase in firing rate. So here in this curve, you can see a contrast of attended and unattended conditions. This increase in firing rate and widening of tuning curve is not the only things that are not only the only effects of attention. For instance, the same author as before, Marlene Cohen, she showed that attention can improve performance also by reducing noise correlations. Well, being very fast and explain what noise correlations is. Basically, when you have one neuron, when you're recording from one neuron and you present the same stimulus many times, you're not going to have exactly the same response as before. So you have a variability in the response. Sometimes the neuron responds more, sometimes the neuron responds less, and then when you plot the tuning curve, you take the average of these responses. Now, you can do this with many cells. In this case, if you take 2 cells, you can see that when the monkey is not attending, there is a correlation in this variability. And then you can see that there is a correlation here. And what attention does is that it decouples the 2 cells, and basically there is a decrease in this correlation of the noises of the neuronal noise, which are the noise correlations. And yeah, you can see a summary of this effect in C. And also you can see that there is a change in oscillatory neurosynchronization in the brain. For instance, in this work here of Pascal Fries, he showed that there is an increase in gamma synchronization and a decrease in low frequencies. But yeah, I don't have time to talk a lot about this. If you want to know more details, please come talk to me later. And also, I forgot to tell, this presentation is in our webpage. So, and all my comments that I wanted to do that would be very more calm are there. So I'm doing just a very short summary because of the time.
Can I ask a question?
Yeah, please.
So I'll produce slides of that. So
Yeah, the way that I understand is that when you are not paying attention, basically the neurons, they are, let's say, working kind of together. And then when you are you start to attend, is that the neurons, they start to become more independent of each other. So there is a reduction in this correlation. Basically, it would be they are working in a similar way. So if you have an increase if you present sometimes a stimulus and this neuron gives a higher response, the other neuron is also going to give a higher response. But and also the same happens. If it gives a low response, the other one is also going to give a lower response. So they're kind of coupled.
So it's because without attention, the tuning curve is just wider, so
Yeah.
they're more likely to
Yeah.
from the same.
This is my interpretation, yes. So now talking a little bit about memory and learning. So I would like to start with the famous case of Henri Molazon. I don't know if I pronounce it right. And basically he was a guy that had many epileptic attacks when he was a kid, and then they tried to treat this with drugs and nothing solved. So at that time there was a new technique that was being accepted, which basically was removal of the focus of epilepsy. And in his case, the epilepsy was so intense that they decided to remove both temporal lobes bilaterally. So they removed basically almost complete the inferior part of the temporal lobe. So here is an original picture, an original MRI of his brain. And here is another MRI of a similar procedure, but this one is made just in one side. So you can see that basically almost all the amygdala hippocampus is removed, and some parts of the entorhinal cortex also. So you throw everything away on both sides. And interestingly, what happened to him is that even though he could still remember the information about his past, like I think it was 5 years until some time in the past, he could not learn new explicit memory. Basically, there was a nurse that would come every day to talk to him, and he would introduce himself every time and say, ask, who are you? So he could not learn anything. And in this picture, you can see more details of the regions that were removed. So 1, 2, 3 are basically these slices of the brain to see how big it was, the removal. And so what we get from this is that these areas, the hippocampal area, is very important for the formation of new declarative memories. And I'm talking about declarative memories because you have different types of long-term memories. For instance, being very summarized again, there is the explicit, which is the declarative memories, basically the memories of facts and events, things that you can tell verbally. But you have also the non-declarative memory, which is the implicit memory. For instance, the memory of riding a bike, or some emotional responses. And for every region, you have every type of memory requires a specific configuration of brain areas. And besides the long-term memory, you have also the short-term memory, which is the working memory, basically a temporal memory of things that you need to be required to do in a short scale of time, like minutes. And for instance, in this experiment, they taught the monkey to fixate in the center of the screen, and then they would show a sample of the monkey, and the monkey, they need to memorize the identity of the object, basically which object was there, and also the spatial location of the object. And then they recorded from neurons in the dorsolateral prefrontal cortex, and then they found object selective neurons and location selective neurons. Basically, the object selective neurons, they would show an increase in activity because this is this phase here, basically, the yellow. And you can see that there is no object there, but you can still see that the neuron keeps the activity related to the sample. So there is a sustained activity during the delay period. And this happens for the identity of the object, but also in the case of the location selective neuron, for the spatial position. And a little bit about learning. So you have also, there is an experiment that was made by the Kandel, the author of this book, that he showed that the habituation and sensitization in aplesia, Basically, what he did was, when you have the siphon in the applysion, and every time you touch the siphon, the applysia has a reflex of retracting the gills. It's a protective reflex. And then what they did was to touch repeated times the siphon and to record from different neurons. And then they showed that there is an habituation, basically an increase. It's changed here. So basically there is a synaptic depression, which is basically a depletion of the readily releasable vesicles. So even though the action potential is shown here, it stays the same, the excitatory postsynaptic potential here decreases. And this happens because there is an increase in the number of vesicles that is released. And the opposite happens for sensitization. You can also increase the amount of vesicles that are released by playing with this neuronal, with the current injections. And so what I just showed you was short-term plasticity, was basically synaptic enhancement and synaptic depression, but you also have long-term plasticity, which is basically, the idea is the same, but while you are, the first one you are playing with the release of vesicles, here you are changing more structural configurations of the cell. For instance, if for long-term potentiation, you can change the numbers of AMPA receptors. Basically, you increase the number of AMPA receptors, and the long-term depression, you decrease it. And so by doing so, you have you can weaken or strengthen the synapse. And well, I didn't time. So basically, I went very briefly through all the core cognitive process, some of the core cognitive processes. I don't have time to go through language emotion, but these topics are going to be discussed with more details during the course. There are specific speakers for that. And that's it. Time to say.